As you can see, I'll be talking about a long-term study of Antirrhinum snapdragons that we've been laboring away on for about 15 years now. And I'm talking about this really as hopefully a welcome relief from all the theory that we've had this week, something very concrete and visual. The slight problem is that this is, I think, a workshop on polygenic adaptation, and I'll be talking about 5 genes, or maybe 6 or 7 genes. So oligogenic, yes. Part of my excuse is that studying a really simple system where we really know the genetic basis will help perhaps understand and illuminate the sort of principles of quantitative genetics that we usually apply to much more obscure genetic systems. Also, I'll be talking about really the inferences that one can make from genetic data, from sequence data. And this will be a bit of a contrast to the main kind of data inference that we've seen, which is sort of GWAS, genome-wide association studies. And one of the themes will be that well, I'm not sure, I was going to say don't collect too much data, but that's not really what I want to say. It's rather that when you really dig into a system, you find that the simple inferences and the simple methods actually can be quite misleading. So this is a system where we have a sort of underlying purpose to really understand a particular population, a particular system, and therefore be able to test indirect methods of inference, which are used very widely, but which usually are not validated against any actual ground truth. It may come across as a bit obsessive in that we're really collecting more and more individuals and really trying to understand the details of the system. But the idea is to find a way to understand a population well enough that we can actually test the rather abstruse theoretical methods that are used for making inferences about selection, dispersal, etc. Okay, so this is a very collaborative project. The work that I'll talk about today is a lot of it with Parvati, who's here, and can correct me if I make any incorrect statements. Not that I will. And the project was largely started by David Field, who was a postdoc at IST quite a while ago, and he's now at Macquarie University in Sydney. We've been, fittingly, I think, for a long-term study, very slow and gradual in publishing, so we have a few papers out, although none of them describing the work that I'll talk about today. And there's a much wider collaboration. In particular, we've had, I should actually didn't put on the slide, but the whole project was really started by Rika Cohen, a developmental geneticist in John Innes Centre in Norwich, who's really working on the molecular mechanisms of pattern determination in flowers. We've been collaborating a lot with Frank Chan, who's now in the University of Groningen, used to be at Tübingen, who developed a neat method, haplotagging, which allows us to get whole genome sequence with a linked read method on very large numbers of plants. That's very recent, so I won't say much about that. Various other people, Daria Shipolina, who's moving, officially, well, always been, but moving back officially to IST soon, currently in Uppsala. Sean Stankowski just moved to Sussex, who's been sort of leading the recent work, ARCA student, and there are many others that I haven't listed. Okay, so a sort of overview of the talk will be that we have a system where differences in flower pattern depend on just a few genes. I say pattern because although the most striking difference between these plants is that they have either magenta flowers or yellow flowers, actually it's not about production of the pigment so much as the distribution of the pigment across the flowers. And it's actually a very rich and complex trait consisting of where the various pigments are distributed. And basically, as has been known for a long time, since work by Fisher and Haldane in the 1930s, 40s, showing that even quite weak selection can maintain sharp differences between populations. So we have a very striking, abrupt disjunction between populations with different flower colors, but that doesn't require any very dramatically strong selection. What's important in this system, in general, is that when we have populations that differ by multiple genes, And when they mix, this admixture generates associations between genes. You rather obviously have an excess of parental combinations. And these associations are referred to technically as linkage disequilibrium. It's very easy to remember the term linkage disequilibrium because it's doubly misleading. You don't need linkage. We'll be looking at statistical correlations between the state of this gene and the state of that gene. If you have an allele from population A at this locus, an allele at population A at this locus, they're more likely to be found together. The state of this locus is correlated with the state of this locus. It's nothing to do with physical linkage, although obviously if they're physically linked, the associations would be broken up more slowly. So in this talk, I'll be talking about linkage disequilibrium between unlinked loci. I'll also be talking about a system that we're assuming is at equilibrium. So that's why we talk about linkage disequilibrium. It's really unlinkage equilibrium. Yeah, no, I don't want to go there. I think other languages may have better terms for this. Anyway, these associations represent a barrier to gene flow, and hybrid zones are often studied in relation to speciation, the idea that a gene, any gene, neutral or selected on one genetic background, in order to get to the other population, has to recombine onto the other genetic background. And if there's selection against the genes it's associated with, it'll move into the other population associated with stuff that is deleterious in that new population. It'll then be reduced in frequency. Effectively, the rate of gene flow will be lower. And this is something that Himani talked about in her talk last week. And so there is a barrier to gene flow represented, really, manifested by these linkage disequilibria, and we can estimate it. It's actually very weak in this system. So this antirrhinum system, I have to confess, is a bit of a disappointment to me because it is apparently, as far as we know, just about flower color differences at a relatively small number of genes. I'll come back to that. So I will have, for nostalgic reasons, a little interlude in the middle going back to one of my favorite organisms, the toads, Bombina variegata, that I used to work on, which are genuinely highly divergent. They're, I think, by any sensible definition, good species in the sense they're very distinct, and yet they hybridize. And so there we really can look at polygenic traits, quantitative traits. So coming back to the antirrhinum, we can use these associations between genes, and we can use the width of Kleins in principle to estimate selection, to estimate dispersal rates, and this is something I started doing with the Bombina. And in this system, because we've been studying the population very intensively, and in particular because we've been building a pedigree showing the relationships between different individuals, we can actually measure selection and dispersal directly, and so we can actually compare the indirect estimates with the direct estimates. And we're kind of in the middle of doing this, and within a matter of weeks, Parvati will have sorted it all out. But at the moment, take everything with a bit of a pinch of salt. And then at the end, I'll come back to this question of whether it really is just about flower color. Would we really know whether there were adaptive differences, selective differences, at very many other traits which we're just not aware of, that we're not seeing, and which are not showing up in the kind of traditional genome scans? So part of this talk will be about both the power and the limitations of the sort of classical population genetic inferences from genetic data. Okay, so snapdragons, these are some of the species, 20-odd taxa, species, subspecies around the Western Mediterranean. Very well studied. They've been a kind of a model system from the first days of rediscovery of Mendelian genetics. In fact, I think Mendel worked on snapdragons, amongst many other things. And in 1900, soon after 1900, there were actually papers on the genetics of flower color in antirrhinum. So it goes back a long way. And slightly, I don't know, embarrassingly encouraging, I quite like it actually, that all this fancy new DNA-based methodology hasn't really discovered any new genes, maybe one new gene. They're all described in the classical genetic literature. Okay. So we'll be focusing on Antirrhinum majus. This somewhat lurid flower, I think, is a domesticated variety. This is Majus pseudomajus, which is one of the subspecies we'll be studying, magenta, and this is striatum, another subspecies. Right?
So what's the main pollination system in these? Is it bumblebees? Is it
Bumblebees, yeah, yeah. It's great big bumblebees. And they're really very sophisticated flowers. They're called snapdragons because they snap open when the bee goes in, and the bee has to work pretty hard to get in there. And in fact, it's sort of on the edge of maybe, I think Mike Lynch should be here because this may be an example of over-design. I'm not sure whether a plant person would agree with this, because it's such hard work for the bee to get in that towards the end of the season they give up, they drill a hole in the bottom, they just take the nectar, and that's that. So this sort of, there's a balance between having a sophisticated flower, which means that only a particular pollinator is going to be able to specialize on that flower, and therefore it will take the pollen to the same species rather than wasting it and sending it everywhere. So you want this specialization, you want it to be attracting a specific kind of pollinator, but you don't want to make it too difficult, otherwise the pollinator gives up. And it's kind of quite dramatic seeing that.
It might be interesting to photograph some of your flowers in UV because the nectar guides that bees keep.
Well, there are nectar guides you can see even without UV. We have done a bit of UV stuff, not very seriously, and it looks kind of not that different, but yeah. I mean, sorry? No, they're not allowed to. This is, I think, quite right and good. I mean, okay, so the wild species have a self-incompatibility locus, and we're actually very interested in that. I won't say much about that today. So it has a very elaborate system, which means that pollen that carries an allele that's shared with the female cannot fertilize. An RNase destroys the pollen. So it seems to be, and we also have a lot of data from the field, there really, as far as we can see, there isn't any selfing. In the greenhouse, you can occasionally see selfing, but apparently certainly less than 1 in a few thousand selfing rate, probably 0. So that's all good. Of course, the domesticated varieties can self, and they have a big deletion in the S locus, which is very annoying because the reference genome is from the domesticated variety and it's missing a big chunk of the locus we're interested in. So, yeah. Okay, so they're self-compatible, they're outcrossing, they're very diverse. As we'll see, they have a high population size. We'll be looking at just the contact between these two, but in fact, these are largely interfertile. So what is a biological species is kind of an open question. Many of these are much more distinct than Pseudomagus and striatum. They can actually have a very different growth form and habitat preference and so on, but nevertheless, they're closely related. Okay, so this is Mediterranean, Pyrenees here, study area. The range of the magenta form, the Pseudomagus, is sort of here and here, rather surrounding the range of the striatum, which is in the eastern Pyrenees. Okay, it looks here as though there's an altitude difference, but there really isn't. In some places, you have the reds at the top of the valley and the yellows at the bottom. In our case, it's the other way around, but you go from one valley to the next, and the geographic relations are different. So there's no real obvious habitat association. We're focusing obsessively on this stretch of road. I don't have a car, so my only driving practice is driving up and down this road in the field season, ferrying the volunteers to their work. Basically, there are 2 parallel roads. Barcelona's down there, you drive up, and you see entirely magenta plants along the road, and then suddenly it switches through a range of sort of hybrid phenotypes, pinks and whites and oranges, into the yellow form. Okay. This, sorry?
Is there some feature that bump, like elevation gain or soil change or something?
Well, I mean, there is an elevation gain, but in other places it's completely reversed. So you can get in fact, with the next valley over, you've got the magentas higher up than the yellows. So there's nothing. We've spent quite a lot of time realizing that it's very hard to measure the environment and describe the environment. They're living along the roadside. There are weedy species. They live in a whole range of habitats, most of them completely different from what happens in the greenhouse. They mostly live not in soil, but in cracks in the rock. One of their favorite habitats is medieval castles. You see these ones you want to sample, right the way up on the battlements, and it's very annoying, but yeah. It's a very, very heterogeneous kind of habitat, but they need disturbance. I should say one of our ethical quandaries is how far to disturb their natural environment by negotiating with the roadsmen who every year after we've sampled and tagged and studied all the plants, they come and mow them down. And, you know, to maintain visibility and so on. We've now negotiated, in fact, Sean negotiated a subcontract, so we now have a range of strimmers and so on, which we can strim round the plants. But of course, in the long run, this is not going to be good for the Antirrhinum, because the reason they were growing alongside the road is that there's disturbance. So in most natural areas, they get overgrown very quickly. Yeah. Anyway, that's the ins and outs of the practicalities. The genetics are pretty straightforward. The main genes affecting the distribution of anthocyanin pigments are Rosea and Eluta, which are two tightly linked genes, half a centimorgan apart. It's been known since 1924, I think, possibly before. And they have opposite dominance relationships, so that in Pseudomajus, Rosea has the dominant allele, whereas Eluta has the recessive allele. That means you can distinguish a heterozygote. There are recombinants, but I won't say much about that. They're quite low frequency, a few percent, 5% or so, consistent with the tight linkage between them. So we'll treat these as basically one locus, determining whether there's magenta across the whole flower versus whether it's concentrated in actually a very small part of the flower. It's usually present but very obscure in the striatum. And then we have the orone pigments, the yellow pigments, controlled by a combination of Favea and Sulfurea. And there's a couple of other genes that we know about that have more obscure effects. And we know where they are on the map. There are 8 chromosomes. Come back to this, and we have markers linked to all of these. So, first of all, we want to know how many genes are affecting flower color. And this is work started by Rico's group. They took 8 samples of about 50 plants each across this transect, the 2 closest pools 2 kilometers apart. And so if you do an FST scan measuring the basically the divergence between populations relative to the variation within populations at each site, or each window rather, these are 10 kb windows. So along each window along the genome, you measure this FST and you see, very nice spikes, and this obvious spike here, which is even colored in red there. This is linkage group 6, this is Rosea and Eluta, and this homes into a megabase region. That's basically the Rosea, there's a couple of coding genes, and Eluta there. Okay. So we have actually a complex pattern that I'll come back to, in which we have a sort of broad increase in FST over a few hundred kb, and these sharp spikes showing an increase in FST. So I'll come back to that. Another way of detecting genes involved in flower color is to ask, do we see narrow clines? And this is a very crude estimate, because at that time, we only had whole genome sequence data from pooled sequences, and only, in fact, from 8 samples, with these 2 kilometers apart, and then about 10 kilometers apart, and then much further apart. So there was, a rather crude transect, but one could nevertheless ask, if we take markers which have a allele frequency difference between the most distant points of more than 90%, so almost diagnostic, and then ask, can we estimate the width of the cline? Basically, we're looking for whether most of that allele frequency difference is concentrated in the 2 nearest regions. This is an alternative measure of divergence, which includes the spatial structure, and it gives similar answers, but not identical. It actually picks up another gene, which actually was already known to be involved in flower color. So combining these two, the FST and the narrow cline scheme, one picks up about 7 genes, which actually, remarkably, were all anyway known to be somehow implicated in pigment control pattern determination. So that's all kind of both straightforward and disappointing. I would like to have had more genes to work with, but there we are. There's an interesting sort of sideline in how one interprets these increases in this FST. As I mentioned before, FST is defined as basically the difference in divergence within these pi. This is sometimes called dxy, but it's basically pi b is the probability if you take 2 genes from different places, from either side of this hybrid zone, they'll have a different SNP versus the same probability for 2 genes from within a population, and you divide that basically by diversity in the population as a whole. And if you just have 2 samples, people sometimes sort of approximate that by saying, well, it's the average of pi between and pi within. We can come up with different formulae, but it comes to the same thing. It's basically a difference between divergence between and diversity within. And as I think was mentioned before, it's pointed out in quite a sobering paper by Cruikshank and Hahn, that actually this means that when you have to be careful in interpreting FST. So initially people had done these kind of genome scans and said these are speciation genes, they're more divergent, they're somehow being kept different by selection. Which may be true, but the increase in FST is actually more often, in fact in all the cases reanalyzed by Cruikshank and Hahn, in those cases it was due to a reduction in diversity. So if diversity within is decreased, then FST will increase. You don't have to have an increase in divergence. It's quite hard to disentangle these. But there are other reasons actually to think that these sharp spikes really have to be due to reductions in local diversity. Because if you ask, let's set up a sharp barrier to gene flow, and in fact this is a very strong barrier because there are 2 loci maintained different by selection. You know, we know that there are sharp clines at these, which we've seen in many different transects. So we set that up, and then you calculate what will be the effect on FST. There's actually very robustly a broad increase in FST over this sort of scale, you know, over a scale actually of order, whatever the selection is. So this broad increase can be explained by the effect of a barrier to gene flow, but it's very hard to, and for like to impossible, to explain these very sharp spikes, whereas sharp spikes are exactly what you expect if there is a selective sweep that takes a few hundred or a few thousand generations and brings to high frequency a particular haplotype. Okay, so we think that these are due to 1 or more sweeps. How many loci are involved? There and there, maybe there. You know, there's probably quite a lot going on in this region. Again, that's something we're trying to disentangle. Okay. So what I'll focus on then is just a very, very small subset of the data, basically looking at 5 markers which are chosen to represent the divergence at the 5 main loci that we have. There's a couple of others, but we're focusing on these 5, and we're treating Rosea and Diluta as one locus because they're tightly linked. So this just keeps things simple. So we can treat all these as unlinked. And we have accumulated an enormous number of individuals. And the reason for that is that we set out to try and set up a pedigree with the aim to make direct measures of dispersal and selection. So we tried to find every plant every year, and I'll come to why we're still doing this after 15 years. But you know, the data is accumulating. We're now up to 30,000 or 32,000, and it's the annoying thing about all this modern molecular work is that it's so efficient and relatively cheap. So we go out into the field, and I usually spend most of the field season driving the volunteers backwards and forwards, and then in between the driving duties, the taxi service, I sit and try and analyze the last 10, 15 years data and make some progress. But then you get back and you know it's quite a few months' work to take the leaves and put them into 96 well plates. You know, it sounds trivial, but you know, it takes a while to do it properly. So we've collected 3,000 or 4,000 samples. We put them all into the plates. We send the plates off. 3 weeks later, the company comes back with the SNP data. So you know, it's just too quick. We don't have time to actually analyze. So we've been accumulating data. We've now made a solemn vow not to genotype any more of the samples. We'll keep sampling, but we're not going to genotype any more until we've analyzed the last.
So Nick, you're just scoring these SNPs, nothing else?
Well, what I'll talk about is the SNP data. So the main bulk of the SNPs is around 100 SNPs on each individual, a mix of sort of boring SNPs for the pedigree reconstruction, and then a smaller subset of SNPs around the flower colour loci. I'm focusing on just 5, which are chosen as representative of those. We've recently, through Frank Chan, got haplotype data on 1,200 individuals, and this is even more annoying than the SNP data in the sense that it's, you know, SNP data is wonderfully clean. I'll show it in a minute.
Yeah, because the haplotypes, you can start doing some of the serious sweep tests.
Yeah. We can do all kinds of things, but there's a lot of heavy bioinformatics, and it's just heavier to deal
What's the
with.
genome size in?
600 megabase.
Okay, so it's quite doable. Okay.
Well, for the SNP genotyping, CASP, so we designed, based on the pool-seq data, we designed primers and it took quite a lot of back and forth, but now we've got actually about 200 that work. Actually, I was really quite surprised and encouraged that, again, it's slightly embarrassing that we've got an enormous number of duplicate genotypes, because we go back, even in the same year, the label might fall off, and then we sample the plant again. And then next year, the label, usually falls off, and so we sample it again. And then it's only when we've got the genotypes in that we know that actually this plant is the same as this plant, is the same as this plant. So to do anything with the data, we have to have the genotypes, actually. But because we've got duplicate genotypes, we can get error rates, and they're 10 to the minus 4 or less, extremely reliable. So that makes it possible to do all sorts of stuff I think you wouldn't be able to do with whole genome sequence type data. Okay. So anyway, we've got this big data set over a long time period. We've got basically a transect which really is 2 parallel roads and a railway line and some various other tracks and so on, but that's the transect. It's kind of convenient because it's more or less one-dimensional, which always makes everything much easier. And for the purposes of the sort of Klein analysis, we divide the 22,000 individuals into about 1,000 demes, where every individual is within 25 meters. And there's a whole palaver deciding why 25 meters, not 10 meters or 50 meters. It doesn't make a lot of difference anyway. And so we've got about 343 demes with at least 10 plants. So, we've got a lot of data. And so the first thing that Parvati did was to use the traditional kind of Klein fitting scheme, where one fits a model. Initially a sort of simple sigmoid model, that doesn't fit very well. You find that there's long tails of integration on either side. You can see that more clearly on a sort of log scale. So we transform from P, going from 0 to 1, to allele frequency ratios, log P over Q. So 1 here corresponds to P over Q is 1, in other words, 50%. 10 corresponds to about 90%, right? Not quite. 0.1 to about 10% and so on. So we're expanding the tails. And on this scale, a sort of simple single locus model will almost always give a straight line. And that's the fit in the middle. And then you see there's alleles characteristic of that. The magentas are on this side. Some of those, a small fraction, a few percent, go even a few kilometers over there. You've got a long tail of integration on either side, but a very sharp change in the middle, over less than a kilometer. And you can see that these Kleins all coincide more or less, but they have different kind of patterns, different widths. But actually, if you look at the data, it doesn't look like this. I have a bit of a gripe about a lot of papers which just show you the fitted Kleins where it all looks rather neat. The actual data here is a sort of cloud of points. And of course, some of this is sampling error, but most of it isn't. There's clearly a lot of patchiness on the yellow flank, on the striatum flank, which you can see in many of these. And maybe it's just that the populations are coming and going. We don't really know. But there's a consistent gradient, a sharp change, and then tails. And if you plot again on this log scale, this logit scale, it's again, who invented this? Fisher. So I think, is that right, Bruce? It must have been Fisher, yes. Used it a lot.
I don't use it early.
Yeah, okay.
We'll see.
Let's say just. Yeah. Sorry? The y-axis, so this y-axis is just the frequency from 0 to 1 of each SNP, right? And this is a transform frequency, right? Log P over Q, yeah. So again, you can see fairly clearly this sort of sharpish change in the middle and then a more gradual change on either side. And actually, a better way or a more convenient way to visualize this, which is used a lot, is to construct a so-called hybrid index, which basically is just to add up the various diagnostic features, in this case, diagnostic SNP frequencies, SNP states. So each individual has a number which is from 0 to 10. We've got 5 markers. They can each have 0, 1, or 2 copies of the pseudomagis allele. So you just add them up. And this is a wonderful thing. It's an additive trait. It's, we assume additive traits, but here we've made an additive trait. So this is sort of one very, very strained kind of connection with quantitative genetics. We are doing quantitative genetics here. And if you just take the mean of this hybrid index, again on a log scale, you see this step pattern very clearly. It's not completely satisfactory, so the analysis we do is based on the individual loci, because if you sort of average a number of stepped patterns which are a little bit different, it's kind of, so one of them does that, and one of them does that, and so on. The overall pattern, if you take the average, will be something like that, and that's a useful visualization, but you actually have a rather broader transition here than you would if you were to look at the things individually. So that's just representing the data using this hybrid index. There's something else we can do which is actually quite convenient, which is to look at the variance of this. So if we take a population in the middle, the individuals each have some value between 0 and 10. So like that. Actually, not far off a Gaussian. The wonder of the central limit theorem says that actually, if you add up a fairly small number of numbers, you often get something close to a Gaussian. And the variance of this hybrid index is expected to be, if they're in Heidelberg and linkage equilibrium, just twice the sum of pi qi, where I is the sum over the 5 loci, right? So this is just because the variance of a 0-1 variable, I think I went through this in one of the tutorials, variance of a 0-1 variable is just p times q. And if we add that up over 10 genes, which are assumed independent, 2 copies of each of 5 loci, we get that prediction. And that is equal to, in fact, what I called in one of the first lectures, the genic variance. So that the additive genetic variance, and all the variance here is additive because the trait is additive, it's defined to be additive. It's actually equal to this plus the sum over all I not equal to j. And if we count both pairs, well, yeah, kind of. Yes, if we just count these d's only once, in other words, this would be, in this case, d12 plus d13 plus d. So we don't double count the d21, then we put a factor of 2 in there. So what we have is that the variance of this hybrid index is necessarily equal to the sum of the variances due to the individual genetic loci, plus the sum of the covariances due to the off-diagonals. There is another term, called it d dashed, I could call it, which is due to the covariance between genes from different parents, so that these are due to covariances between, draw it up here. Okay, this represents, a pair of genes on 2 chromosomes. And what we're doing is saying we can divide up the variance in the total trait, which is the sum of contributions of all these different genes, into a component due to the gene, the variance contributed by the genes individually. And if they're independent, that's everything. But then we also have this linkage disequilibrium term, which is due to associations between genes inherited from the same parent. We also have this term here, which is the correlation between the genes from the 2 different parents. Okay? If we have random mating, random union of gametes, then this should be 0. And actually, it's quite low in the Snapdragon case. So we have these terms, genic variance plus these basically LD variants. What would the term for this be, Bruce?
Yeah,
It's not quite the same. Identity, yeah, anyway, we won't get into this. Okay, we've got these components.
I think what's called
Yeah, you can call it that. Anyway, I'm going to ignore it. So the point is that it's actually rather easy to take your sample, calculate your hybrid index, calculate its variance, and then subtract off this, because you know what the allele frequencies are. So the difference between those, that excess, is due to linkage disequilibrium, okay, basically. And this excess is expressed here as a correlation. The way you do that is to basically take you divide by the product of the variances, right? So, there's different ins and outs of exactly how you do this, but it's a fairly straightforward way of estimating the underlying linkage disequilibrium. The alternative is to do maximum likelihood estimation on every pair separately, which we've also done, and it gives similar answers, but it's just more of a pain. Anyway, you do that and you find there is an excess. This is a little bit misleading because there's a lot of noise in there. The actual average here is about 0.03. It's not that high, but it's very consistent and very significant. So there is an excess association even between unlinked genes, which we think is most likely due to just mixing. You really can't avoid having some excess LD due to mixing. Okay. So this is the very slight connection with quantitative genetics. This is quantitative genetics, it's just only marginally polygenic, it's by loci. So this is where we get to in the first phase. We have clines that coincide, which have widths that vary. The narrowest is at Rosea and Diluta, about 750 meters, Sulfuria, the next one. And the widths actually correspond with what you'd expect from how dramatic the effects of the loci are. Now, there's very robust theory going back to Haldane and Fisher, really, that if you have a balance between dispersal, represented by diffusion of genes with a standard deviation of sigma per generation, so sigma is a measure of the dispersal range in the generation, that's balanced against some kind of selection. And I'm going to be quite agnostic about what the selection is. All you have to have is selection that favors the common allele here and the rare allele here. It could be selection against heterozygotes, it could be selection against certain epistatic combinations. If only the combination of alleles found in magenta is good, combination of alleles found in the yellows is good, then selection will act against the recombinants. That will also maintain divergence. Or you can have frequency dependence. If the selection is mediated by bees, and if the bees simply train on whatever color is common, then in the yellow side, they go for the yellows, and they don't know what these weird magenta things are, so there's a disadvantage. This is called positive frequency dependent selection. So we have all kinds of selection. Maybe all of these are going on in this system, actually. But as far as the theory goes, it doesn't really matter. There's just a measure of selection coefficient, basically with the net selection against alleles on this side and for alleles on this side. And that predicts the width. So, we're done, right? If we knew what the dispersal was, then we could predict the selection. If it's 100 meters, then it's 14%. If it's 50 meters, then it's 3.5%, so it's not so easy measuring dispersal, but we'll get to that. But we can do more than that, actually, because if we assume that these correlations between loci, these linkage disequilibria, are due to mixing, we actually know the recombination rate is 50%, they're unlinked. So for unlinked loci, given some value of R, we can predict what dispersal rate is needed to generate that. And in this case, it's around 50 meters, so then we say, selection's about 3.5%. So we've got this measure purely from genetic data, right? Don't have to do any difficult work actually following bees and stuff. So that's all fine. And this is, an approach that's been quite widely used, I think was popularized by John Endler in the 70s.
So Nick, just a quick question. You don't know about the pollination biology, because it seems that the dispersal are the bees.
Yes.
So
well, and also seeds, right? So there's two things.
Sure.
There's pollen moving and then the seeds move, both.
The seeds don't have a filament to float very far, right?
Well, we'll get to that.
Okay.
They're a mystery.
I'll hold off then.
Yeah. Okay. So we asked now, can we explain the width of these various clines and this stepped pattern through some simple model with selection on 5 unlinked loci? plus some dispersal, right? So we'll get back to that. But I now want to have an interlude with toads, okay? Really, because this is a very contrasting system that I started on in the early 80s. In fact, I remember in 1981, I think, getting a big parcel of rather tatty yellowing paper, sort of photocopy paper, because it wasn't even photocopy paper, because in Poland at that time weren't allowed access to photocopiers. So Jacek Smura, who'd done this heroic amount of work on these two species of toad, had typed out all the Allozyme stuff, and it was difficult to do the work at the time. Also, getting supplies. So he made his own starch for the gels by boiling up potatoes. So it's but I like this. It's a low-tech method, and he did a really big survey with 2,000-3,000 toads genotyped at 6 markers, right? And in fact, with the advent of DNA-based markers, there was a long period of maybe 20 years when sample sizes dropped, and Klein analyses got rather pathetic because you couldn't afford to do big samples. And now they're creeping up again, but still, Allozymes, if you want a large sample, those were the probably in many cases still are a relatively cheap way to go. Okay, so these are the toads. They're really different. The Bombina bombina, in German called Unken. There's a novel. Anyone remember the novel by? It slipped my mind. Anyway, the Unken is a prophet of doom. So it's a very gloomy novel, I think, because they live in large permanent ponds, and in the evening they'll be calling, rum, rum, and it's quite extraordinary, very eerie, and at dusk they have this booming sound. So they're adapted to this sort of permanent water. They spend most of the time in the water. They have large vocal sacs. They sing in large choruses. They lay larger numbers of quite small eggs, which then develop, more slowly. Whereas the variegata lives in upland, hilly areas in temporary puddles, little puddles left by tractor wheel ruts. I don't know what they did before tractors, maybe horse and cart, but they needed ruts in the ground to produce these puddles. And so they're more terrestrial. They have thicker skin. They have longer legs. They lay larger eggs. The tadpoles are less shy, actually, because there are fewer predators in these puddles. The Bombina bombina here have tadpoles that are really quite shy and will react to predators, etc. Everything you look at is different. DNA content is different. Genome size is different. It mostly repeats. They are estimated to have diverged 5-6 million years ago, very distinct. But they hybridize perfectly freely, apparently. So if you go round the Danube Basin here, where you find Bombina, or around the northern European plains, then you find hybrid zones all the way around the Carpathians, all the way around sort of the edge of the Balkans here. And so the early work we did with Jacek was here in southern Poland, and then I did field work over a number of years in Croatia there. So it's a very neat system. And this hybrid zone is really only about 6 kilometers wide. So it's a very striking distribution. So you look at these Allozyme markers. These are the 6 markers from Jacek's data from southern Poland. And you can't really see the symbols. There are crosses and circles from 2 different transects, which amazingly are basically the same. So I really, it's amazing that this is very replicable. Of course, if you go to other places, it's different. But these 2 southern Polish populations gave very similar patterns. And again, this is on this log scale. So you see the stepped pattern with a sharp climb about 6 kilometers wide. And again, strong LD, much stronger than in the Snapdragons, about 0.22, right, reflecting stronger selation and stronger dispersal. And so you do all sorts of inference from this. If you assume a sort of diffusive dispersal, you can estimate it to be about 1 kilometer in a generation. Difficult to measure directly. These toads live up to, the record was 30 years, I think, mark them. They're nice because this warning pattern, which they display if you annoy them, is unique to each individual. So you photograph them, you have a book, you look in the book, and you can identify the individuals. Drives you mad, but you can do it. Okay, so you can estimate the selection out here on individual alleles, because out at the edges, LD is broken down. Doesn't need to be very strong. These are long tails of introgression. But in the middle, there's a sharp step, and you estimate that the total loss of fitness has to be quite substantial. The mean fitness in the middle has to be about 60% of the mean fitness on either side. And you have to have a lot of loci in the selection. So you do all sorts of fun population genetics. Just want to mention here, though, the quantitative genetic angle. So this was on the Croatian transect, which is different in some ways. But as Beata Nürnberger was doing a lot of this work, we measured 6 different traits. So belly pattern, this is a kind of fun trait. If you go back to, it's an idea Jacek had, and we actually used it mainly to identify individuals. So you can describe the belly pattern. You look at how do you describe that? Well, there are certain points there and there and there and there, so on, which always have color. And then you just say, are they connected or not? So you have a binary score, series of noughts and ones, and I think it's a 10-digit binary score, 10 on the left and 10 on the right, something like that. So you've got a large number of combinations, and that means you have your photo book, and each page is a particular binary score, so you only have to look through 20 toads rather than 2,000 toads. So you do that, but actually you can just add up that number, and it gives you some kind of quantitative measure of the amount of color. We did do a bit of photography, but I think this spot pattern is very easy and seems to work pretty well. Anyway, that's maybe one of the more peculiar things, the belly pattern score. This is the cycle length, that's a mating call. They have different frequencies of call. So that describes the call of the males. Skeletal proportions, we actually, so another, yeah. So you anesthetize the toads, they lie flat on their backs for a few minutes. And we would X-ray them, but we X-rayed them in the local vet. And so it was always a little bit tricky making sure they were anesthetized and didn't wake up in the middle. But we got quite a few, several, I think about 600 X-rays. So measure the skeletal proportions. This is a principal component, or sorry, a discriminant score. Discriminant score is a measure combining all the various skeletal proportions. Egg volume, straightforward. Skin thickness, straightforward. Development time of tadpoles, and so on. Now, what's striking is that 4 of these are not significantly different from linear. There, and there. They're actually pretty much linear with this, which is an allozyme hybrid index. So I should have said, this is the quantitative trait on the vertical axis. On this axis, the 6 allozyme markers, okay? Going from 0 to 1. So this was actually deeply disappointing because the point of doing this was to see which traits are under selection. The allozymes, well, of course, there was a heated debate then about whether they were selected or not, but one would imagine they're not super strongly selected, and there were these estimates, indirect estimates, maybe a few percent. But then, traits like skin thickness and mating call, you think they'd really matter, and they'd be sharper clines, but they're not sharper clines. There are 2, egg volume is somewhat displaced, and development time is somewhat displaced, but the overall picture is that everything changes together. The other thing that's neat, again, goes back to this different components of genetic variants. So if you look at the top diagonal, this is taking an enzyme score, a hybrid index, all right? And for some reason, we only used 4 of the loci, so it goes from 0 to 8, whatever. And then we've got various things here. These differences are the difference between the 2 parental populations, the various traits. And you can measure the covariance between these different traits in the pure Bombina, the hybrids, and the variegata. And you see very consistently, all of these have a strong excess, well, maybe not there, all right, quite consistently. You see a strong excess covariance, which is seen in the hybrid populations. Now, you could say this is pleiotropy. You can always interpret covariance between traits as some sort of shared genetic basis. But I find it really hard to imagine that there's sort of strong pleiotropy between all these completely different traits. Right? So then there's actually a very neat relation which allows you to estimate from the covariance, from the excess covariance generated by LD, the underlying linkage to equilibrium. It's basically the covariance divided by the difference in means for the traits between the 2 sides. And that's something which is dimensionless. So you take the covariance between 2 traits and you divide by the difference in means of the first trait, difference in means of the second trait, and you get a number which is basically giving you an estimate of the LD, which is down below the diagonal, okay? And so these estimates are actually I didn't show the table, are reasonably consistent if we calculate, I should have put in, I don't think I put it in, if you sort of take the average for each trait and measure the mean LD, it's a bit all over the place for each pair, but the average really comes out pretty consistently. So the point of all this is, we've got all these different traits, we've got this complicated system, but if we look at the covariance between traits, we can predict it very well just from the LD that we see between neutral markers. So this is kind of surprising to me. It's because it says that, well, in all these different traits, there's no evidence of effects, differential effects of selection on these particular traits. No doubt there is selection on everything, but it seems to spill over. So it's all a matter of collective selection on everything in linkage disequilibrium. And that actually is the explanation, I think, for these sharp steps that we see here. Because what's happening is that in the middle, the selection on a particular allele isn't due to its effects via particular traits. And even the selection on particular traits is not due to that trait per se, but it's due to everything that's all associated. And there's strong associations. R is 0.22. It's quite a strong correlation, which dominates and means there's this sudden sharp step which reflects a collective selection. So we actually now come back to snapdragons. What's happened?
Can I ask something?
Only when I've managed to rescue this. Yes, sorry.
So when you say that you were surprised to find these phenotypic clients to be flat or linear, isn't that an indication that maybe you haven't gone far enough sampling and then it would sort of plateau at some point?
It's not geographic flatness, right? So, yes. So, sorry, what were you saying was that we're not going far enough out?
You were commenting on the fact that the phenotypes co-varied perfectly with the allozyme frequency, but I'm not sure I understood very well what the x-axis was, so I thought maybe if you sample further out.
Then they should become decoupled, that's right.
Yeah.
Yes, because the LD should break down. In fact, we do see that the LD between enzyme markers, which we can measure further out, does break down. It sort of has to, because you're imagining a set of genes coming in here, and they're sort of together, but recombinant every generation of dispersal, which is a kilometre, another kilometre, there's a round of recombination and everything decreases by half. So things should be uncorrelated. We couldn't measure that for the phenotypic traits because, you can't measure them when they're not very variable, when they are basically more or less one pattern or the other pattern. It gets very noisy. You can do it for the enzyme markers because the Mendelian segregation it's a more clear-cut trait.
So you have a hopping stone model instead of
Yeah, it's a hopping stone model, exactly. Okay. We did do some sort of direct, so we were marking all these, we weren't marking them, the toads had marked themselves, so we could follow individuals. And we did find this one toad, Jacek found this, which he'd been to Croatia in the 80s, and then in the 70s, I think. And then we went back, and it was 28 years later, and there was this toad. And so we had some dispersal estimates, which were in the right ballpark, but obviously you can't really find enough to get good estimates. Anyway. Yeah.
So just to make sure I understand, if you're saying it's really these associations, this correlation between loci that's causing this sharp climb at each locus, why doesn't it cause the same thing for the phenotype? This is what if you have this, you were saying there's this collective effect that should.
Well, actually, it does in a sense.
But because you raised the point that the phenotypes are much more linear than you.
Yeah. So the linearity is actually showing that it's linear, that the phenotypes map linearly onto the allele frequency.
Oh, allele frequency.
The allele frequencies have a step, and therefore the phenotypes have a step as well.
Okay, I had missed that.
It's just that you can't estimate a step directly very well for phenotypes because it's noisy at the tails. You've got more sampling error. So I think I did look at it, but it seemed to make more sense to look at.
Yeah, but I thought it was distance. Sorry.
No, it's not distance. It's allele frequency. Yeah. So otherwise, you're exactly right. Yeah. Okay. So we go back to the snapdragons. Can we explain what we see through a simple selection dispersal? And as you'd expect, the answer is. What's the answer? It's complicated. This is a good.
It depends.
Yeah. Well, I think it's complicatable. Anyway, so what we have, we've got this panel of 98 SNPs that we are using for building a pedigree. And the blue dots are the SNPs. There's 8 chromosomes. The red dots are the 5 markers on different chromosomes that we're using. That's Rosea and Diluta very close together. That green is the self-incompatibility locus, which is a whole other side of the project I won't talk about. And these are the data. And it's kind of a bit sobering that these are the data. I can't remember, it costs quite a lot of money for it. But anyway, that's it.
You just need to hit the side of the TV in the picture.
Yeah, exactly. So, here we have the genome. So we go from the S locus, which is on chromosome 8, and then I can't remember which one is up on chromosome 1. This is Rosea Diluta. It's all non-linear because we had a lot of markers, about 25 markers around Rosea and Diluta. That's this set of markers here. And you can see this is distance, so they change. That's the decline, right? And you can see that one's changing, right? Less dramatic. That one's changing a bit. Again, distance is non-linear because all we do is line up the 22,000 individuals in order of where they are on the transect, and most of them we're sampling the middle because that's where the action is, right? So this is the central kilometer. This is the central 2 kilometers, basically. And then there's, we have a decent sample size out there, but we have an enormous sample size in the middle. And then, sorry, the colors are white, gray, black, homozygote, heterozygote, homozygote. So those of you with good eyesight can just read down this column and see that this individual and this individual are likely to be the parent of this individual, right? So if you've got good eyesight, you can reconstruct the pedigree from this. But maybe smartphones will be able to do this one day. I don't know. No one will put the investment into the app. Anyway, that's the data. But it sort of shows that you can spend years analyzing even this relatively small bit of data. I mean, whole genome sequence is, for humans, it's 3 billion. So we've got 3 billion down there, and we've got, tens of billions of SNPs segregating. So, even a modest SNP chip has, orders of magnitude, 3 orders of magnitude more SNP data than we have. We do have more individuals, so they're ahead on that. Anyway, what do we do with that? Well, so David has been sort of using a program called Snippet to just infer relationships. And we're really only at the beginning of this, so he's been quite conservative, just saying, let's identify offspring where we can find both parents, so trios. So there's lots of other information we can get to, and we hope to get to soon, but for the moment, there are, out of this big data set, 2,400 trios that we've inferred. And you might realize that that's not all of the individuals by any means, because a lot of this is that we don't sample every individual. We're sampling about 60% of the population, probably. Also because parents will be outside the study, either before the study or outside geographically. We still haven't really produced a convincing calculation that understands why it is we pick up basically about 10% of individuals we can assign both parents. Yeah.
So do you sample the same individuals at different years, or can you?
We do by accident. We try. If it's still got its label, we don't bother genotyping. But if it's lost its label, we will genotype it. Yes. So we have a lot of data on the life history. I'll come to that actually. Yeah. So and again, it's an interesting story. I was told it was an annual plant when I started. Therefore, we had a 4-year grant, and that would have been enough to have 4 generations. But it's not like that. So you can see that here. So this is taking all the 2010 adults, which had been assigned as parents, and asking where are the offspring. So here are, for parents in 2010, some of the offspring were inferred as 2016. That's 6 years on. So this is geography. This is basically the transects along here, and there are an enormous number of trios. This is a little bit misleading because you can't quite see. Mostly, this offspring will have 2 parents very nearby. Sorry. Yeah. So most of the dispersal is very localized, but what stands out when you look at these plots is these sort of dramatic movements. They're actually a minority, so we'll come on to that.
Could it be seed dormancy?
Yes, exactly. Although,
Can you go back? It looks like that one parent seems to have basically all lost in 2016. But this one, second,
yes, yeah.
So how can you explain this? Is it
It's all weird, actually. Yes, yes. Well, we'll come. So what's going on here? Well, if we summarize all the data, this shows if you take individuals assigned as parents in 2009, their offspring were, 3-4 years later, etc. Of course, the 2015 adults can only have offspring in 2016 because this is an analysis of the up to 2016. We've now got more data, and we haven't yet analyzed it, but basically there's some kind of dormancy of 3 or 4 years. And I'd always, as you said, oh, it's the seed bank. The seeds drop down and they just sit annoyingly waiting till we've gone away, I suppose. And we can't exclude there being a 50-year seed bank and then we're doomed, but whatever. Probably 3 or 4 years, yeah. But it's weird because if you put them in the greenhouse, they all germinate. So I have a great student just started who pointed out we don't know it's a seed bank. It could be that they germinate into little seedlings, and we don't count little seedlings because it's maddening. You can't really find them and you can't identify them and so on. So she's actually going out next week to see what happens. She just did the simple thing of scattering some seed and seeing what comes up. So there's dormancy, maybe a seed bank, maybe not. Yeah, this is why we're still doing it 15 years later. Because everything is 4 times slower, but the sample size is 4 times bigger. So, yeah. Okay, so we can get at this we get a dispersal here. The pollen dispersal is just the distance between the 2 parents, between the mother and the father. That is pollen dispersal. And that's the green line. And I plotted this because I like doing scale transforms. Scale transforms are wonderful. Firstly, log of distance. So this is 1 meter, 10 meters, 100 meters, 1,000 meters, distance between the parents. Down here, it's all measurement error. I mean, yeah, we don't really measure them, the plant location to within more than that much. Up here is a probit transform, which means that we've used normal distribution to define basically 0 corresponds to 50%, 1 standard deviation, 2 standard deviations corresponds to plus 97.5%, minus 2 standard deviations, 2.5%. So we're taking the cumulative distribution and we're stretching it out. And the reason we're doing this is that a straight line is a log normal. So the actual dispersal distribution, pollen dispersal distribution, actually fits a log normal rather well. 1, 10. So the median is about 18 meters, right? So that's where this green line cuts there. It's about 20 meters or so. And that says that half the pollen is moving less than 18 meters. So the bees move further than that. They probably have a range of 50 meters or something, but they go from one plant to another. So they're mostly taking the pollen to a neighboring plant. And then, the plus or minus 2 standard deviations is something like, something trivially small, less than 1 meter up to a few hundred meters. So the sketch would be something like that. That's the log normal distribution of the distance between the parents, which is the pollen dispersal distance. That's the green. Seed dispersal is a bit tricky because we don't know who's the mother and who's the father. We don't have any chloroplast or mitochondrial markers. And so there's different ways to do it. You could say, well, the nearest parent is the mother because seed doesn't go as far as pollen, maybe. You could do it actually a bit better by saying, well, let's take those trios where the parents are close together, right? And then it doesn't matter which you choose as the mother or the father. But you can do a maximum likelihood estimate assuming that pollen, seed dispersal, and seed dispersal are independent, which is kind of reasonable. So they all give the same answer, basically. The red is probably the best, the maximum likelihood. And the median seed dispersal is about 3.5 meters. So they're really not going far. But some of the pollen is going hundreds of meters, and some of the seeds are going hundreds of meters. In fact, the seed dispersal sort of bends over. So there's more of a tail of seed dispersal. And we also have really unlikely individuals, magenta plants found 2-3 kilometers away from the middle, yellow plants 2-3 kilometers away from the middle. And we've got enough markers, we're pretty confident that those really are long-range seed dispersers. Seed dispersers because they actually have the magenta parental genotype. We don't get many F1 genotypes. So, we've got a few percent, 1-2% of long-range dispersal going even beyond 1,000 meters. This is simply set by, the study size. So the pedigree is within about a kilometer range, so we can't really measure anything beyond that, but we can get these long-range dispersers. So we think that it sort of extrapolates out somehow. It's obviously less certain.
Sorry, I just couldn't understand. What is the variance in the y-axis? You said it's deviation from...
This is measured in standard deviations on the normal. So it's a cumulative distribution, which would so if we did it on the usual scale of distance 0, this is radial distance, 0 to 1,000 meters, then the distribution would be, something like that.
It's the distance of the offspring compared to the geographic.
So the green line is the distance between the mother and the father. So that's the distance the pollen is going.
Between the mother and the father.
So we've got trios. So we have two things. One is the green line, which is the distance between the mother and the father, which is the pollen from the father to the mother. And then the seed dispersal, we don't know because we don't know who the mother was, but we can kind of infer, assuming the pollen dispersal is independent of the seed dispersal. Various methods, but they all come out something like this curve, where seed dispersal is only a few meters, right, which you'd expect. They don't have any obvious dispersal mechanism, but some of them go a long way. And we don't know what that is, wind or something. Yes.
And you never had in your data something indicating that one of the parents was from a different year? I mean, it always matched what you expected?
Now you're getting into all the tricky things, which we haven't, I think, got to the bottom of yet. So, of course, mostly, the parents have to be in the same year, I think, biologically, that's it. But of course, some of them live longer. So they were supposed to be annual, but this was untrue. Not only is there a seed bank, but about half the plants live for more than 1 year. And then half the plants that survive 2 years survive to the 3rd year, and half those. So it drops off pretty quickly, but there are plants, which actually relatively bigger plants probably, which live a few years. So some of the parents inferred in these trios are measured in different years, but we also know that we'll have a fair chance of missing one or other of them. And also some of them are measured in 2, 3, 4 years. So we don't know when they were reproducing. But, so I think I haven't yet got to a statistically optimal way to estimate pairs of parents, given the information you have about the chance of seeing them and given the constraint they have to have been in the same year. Yeah. So it's if you like statistics, this is really good. Yeah. Okay. So we've got the dispersal, and so I'm just taking that dispersal as kind of given. I know it's a really good estimate, probably, although the tails are rather uncertain. So then you say, well, what do we expect to see? And so I think this is beyond any sort of theoretical analysis. It's a fairly straightforward model, but part of the trouble is that there's actually an infinity of models you could choose because you've got at least 5 loci, and you attribute fitnesses to all the different things. I think there's 243 different diploid genotypes, and of course their fitnesses might depend on the frequencies of the 243 genotypes in the same thing. So you've got some vast array of fossil models, and we do think there might be frequency-dependent selection and all kinds of things going on. So can't deal with that. So instead, traditionally in population genetics, in modeling, one takes the simplest possible model, and I think one can kind of justify it because models of dispersal selection balance are usually pretty robust. What matters is the rate at which an allele decreases on one side and the rate at which it increases on the other side. So we simply assign a model which has S0 on one side and S1 on the other side. And that's essentially selection against heterozygotes, although positive frequency-dependent selection is essentially the same thing. What matters is that you've got some selection coefficient, forcing the certain allele down on one side and up on the other side at these two rates. We also simplify things by only looking at the deterministic component, so we're not putting in drift. If you did individual-based simulations, which of course one could do, then you'd have to do zillions of replicates, the whole thing gets to be much more complicated. It's a big population, a big sample, so I think it's justified to ignore drift. So we just track haplotype frequencies. Again, we're missing out details where we might need to follow diplotype frequencies, but we're keeping it very simple. So we're just following the frequencies of the genotypes in the gamete pool. And when we fit the data, we don't fit to the full genotype frequencies, which would again be a statistical nightmare, I think. We just fit the allele frequencies. But that gives us a cross-check, because at the end I can say, well, given this fit to the allele frequencies, does it explain other features of the data? And we do allow for random fluctuation, so the sample size is quite big and it varies across the transect. So we do have to allow for the fact that at any point, the allele frequencies are fluctuating partly by sampling error, but also there's a true underlying variance described by a kind of FST. FST is measuring the variance of allele frequency around our best fit. And then the final point, which is kind of a bit tricky to deal with, is that if you set up a model with S0 different from S1, the Klein trundles are off in favor of one or other allele. And in general, we have a model here with selection against heterozygotes or frequency dependence. We're not putting in any environmental difference. So in general, these things will move. And what we do is assume that there is some divine intervention which pushes it back. And that could consist of an environmental gradient of some sort. It could consist of a density gradient. It could consist of local dispersal differences. We just assume that something is keeping it in a certain spot. Of course, it could be moving. It can't move very fast because it's only a limited transect, and we know that it would fall off the edge of the transect if it moved too fast. Anyway, that's a sort of very rough outline of the simulation. So if we start with really simple simulation, take one locus, just simulate one locus, selection against heterozygotes, symmetric selection, 2% selection. We ignore the tails of long-range dispersal for the moment, this black curve, and we fit for ROS, and actually this quite weak selection sort of fits, but it's actually too broad in the middle and too shallow in the edge. If you look on the log scale, you can see that it's really not working here, and the reason it's being pulled broader is that there are some long-range things out here, and so it has to have weak selection to fit the introgression. If you put stronger selection in, of course, you can get it to this blue curve now, you can get it to fit in the middle, but now it fits really badly on the edges. So it's actually less good fit. You've got this tension with a step climb. If you fit a sort of simple one locus model like this, even with the leptokurtic dispersal we have, the leptokurtic dispersal, I should say, when I say no tails, I mean just what's observed on this, just what's observed from the pedigree. We're pretty confident on that, but not including these individuals. Right? So we put in this observed dispersal from the pedigree, and we put in some sort of selection, single locus, and we're not really getting a very good fit. If we put in the long-range dispersal, these tails, we get a dramatically better fit. And the reason is that actually we can now fit, with red, we fit these long-range individuals, which are, pulling the thing out. The stepped pattern really shows up strongly when we allow for this leptokurtic dispersal. So I should have said that this step pattern, which is very clear in the data, actually can have two explanations. And the explanation I'd been fond of, because it involves fun theory, and which sort of seems to work in the Bombina, is that it's due to linked selection. That in the middle, you've got all the selection acting together, producing a strong step. That was our interpretation in the Bombina. And that is going on here because we do have LD, and therefore we do have an effect of linked selection. But in the simulations, there's a much stronger effect of this long-range dispersal. Okay? So now we go on, and we do simulations with 5 loci, because we know there's selection on 5 loci. They're not linked, but we know there's LD, so they do influence each other, and they influence each other in the simulation. And so I do 3 things. I do, first of all, compare back to the individual loci, throwing away the LD, simulating loci one by one. I do multiple loci, including the LD, with symmetric selection, multiple loci with asymmetric selection. And this asymmetric selection allows the clients to be jiggling about to a slightly different location. And so the fits for Razier aren't that different going from individual loci to multiple loci. The reason is that Razier is under strong selection. It's a sharp climb. So the effects of the other weakly selected things aren't as strong. But when we look at something like chromosa, there are big differences between the individual lockers, the black and the blue and the red. Chromosa doesn't fit terribly well, but the best fit is something like this red thing. And when we put everything together and try and get a best fit across all 5 loci, this is assuming linkage equilibrium, doing the loci one at a time. This is symmetric with LD, asymmetric with LD, and the best fit overall, kind of either this or this, but this has many fewer parameters because in order to fit the loci one at a time, you've got to get them all to stay in the same place. And actually, that kind of is a bit peculiar because with these models, they would actually be kind of moving. So anyway. Sorry? I don't, I was going to, you didn't get to it yesterday because we could have had an argument. It's like, I don't believe in AIC. Yeah. You can, yes, but I think it's a choice one has to make about how to trade off likelihoods, which this is the total log likelihood, against degrees of freedom. The thing is that we've got, I don't know, 5 more degrees of freedom here than here, so it will give a lower log likelihood.
Yeah, and it's just like adjusted R-squared, AIC and BIC trade.
They trade in different ways, but I don't see why you choose one or the other. This is mysterious.
Well, they often rank things the same.
Sorry?
AIC and BIC often rank models in the same way.
Yes, but there are other ways to trade off anyway, yeah. I mean, I'm not super confident about taking these too seriously because there's a lot of noise around which may not be captured. And there's an attempt to capture it with this FST, and these FST are not actually so big. The best fits, again, a measure of fit is that the FST required is 0.07, similar for those two. That's twice as big as the FST for the so-called neutral markers, which are at 0.03 locally. So, I mean, again, I said we're in the middle of this, so I wouldn't, bet very much any money actually on these being the right answer. But we're getting selection coefficients of, modest, believable, I suppose. I don't know. And then what I'm surprised at, and I'm relying on no one having a good enough memory to remember these estimates from one slide to the next, because we have, sorry, before I get onto that, I should just say, we do have another cross-check, which is these 5 locker simulations do include, they predict the LD. And of course, we've measured the LD. So if we look here across the transect in distance 10 kilometers, 15 kilometers, 20 kilometers, red is the sort of hybrid index, the average allele frequency, with its sort of step shape. The blue is the observed LD, measured at this R coefficient. So that's a sort of correlation coefficient. It's a bit all over the place, but it's sort of appreciable. This is about, I can put the scale, and that's 0.03 there. And it's actually higher in the flanks because of these long-range tails. And then the prediction from the simulations, the best fit simulation from this line, is similar pattern, a bit higher. And actually, I think the discrepancy here may be because one has to make choices about the life cycle. And I haven't even got my head around it. I think Parvati can sort this out because it's a bit mind-bending. How do you actually decide when is selection acting? I mean, we sort of know this, but one needs to come up with a more sophisticated simulation that allows for the fact that selection is probably acting on pollination success. But we haven't got to that yet. Anyway, so final slide. Direct estimates of fitness. This is the point of the whole project, which we've only gradually got to, and we've hardly started. This is a very preliminary attempt, fitting a model of frequency-dependent selection, where we are now asking how many offspring does an individual have. Now, this is really quite tricky, because what we have is this rather incomplete pedigree with a whole lot of trios assigned from a subset of individuals. So we can't really say, how many offspring did this individual have? Because it entirely depends on where it was and when we sampled it and this and that. So it's really not a good measure of fitness. What we can do is say, for any individual assigned as a parent, what's its genotype? And then we say, look at its neighbor, what's its genotype? So the neighbor should have the same chance a priori of being assigned as a parent. But we can ask, what's its genotype at the SNP, right? At the, a particular SNP, let's say Rosea. Here. And so we can ask if there's a systematic difference in genotype between the individuals actually assigned as parents and the individuals nearby who might have been assigned but weren't. So we fit then this model where we say that this P is just the average frequency of these 5 semi-diagnostic markers across the transect. I mean, one could argue about what to do with that, but it's basically says that on the left-hand side, there's selection S0 against the allele. Or sorry, S0 actually, it's signed so S0. If S0 is negative, that's against the rare allele. On the right-hand side, if S1 is positive, it's in favor of the common allele. And amazingly to me, the estimates all come out for all the loci, minus, plus, minus, plus, minus, plus, minus, plus. And these are estimates of selection which should be comparable with the previous estimates, but I'm relying on no one remembering what those previous estimates were. So all of these are very tentative, so I wouldn't, they're in the same ballpark. I wouldn't say more than that. You know, Rosier is stronger than the other ones on the whole. These other measures, sigma is the standard deviation of the estimate across permutations, where you do it, where you make the same estimate, but sort of choosing random individuals rather than the assigned parent. That's the number of standard deviations against a permutation test, and that's a p-value in the permutation test, without doing any of the extreme value theorem extrapolation that Bruce was talking about. It's just very naive. So, you know, these are significant. If it's close to 1 up here, that means it's highly significant. Where are we? If it's close to 0, it's significant in the other direction, etc. So those are not significant. That one is almost, you know, conventionally, those are, you know, marginally significant. So that's the general pattern. Rosier, it's very clear, and it's the sharpest Klein. The other ones, it goes in the right direction. So that's sort of, you know, it all seems to make sense, and we hope it will carry on making sense. And we will try and avoid this sin, which we alluded to in the discussion yesterday, of I wouldn't go so far as saying p-hacking, but you know, there is a danger that you carry on analyzing until everything is a beautiful story, and then you publish it. And you know, how do we control for this? If we carry on analyzing, it'll get more complicated. Where's the optimal place to stop? We hope we'll publish where we are now. It's been long enough. Finally, summary. We've done genome scans, both looking at excess FST and sharp Kleins. Those find flower color loci. Kind of not surprising. We've got 2 populations. They differ in flower color. We find flower color loci. Very simple genetic basis involving, you know, of order 7 genes. Come back to that. We have associations, linkage to equilibria, even between unlinked genes representing a genetic barrier. We have very clearly stepped Kleins, and those could be due to either to long-range dispersal or to linked selection. And the simulations we've done show that mainly this step pattern is due to this long-tail dispersal, which is really extreme in these plants. Very, very electrochotic. There's a whole lot of kinds of selection could be going on: dominance, epistasis, frequency-dependent selection. I suspect they'll be extremely hard to distinguish, but you know, we can try. The problem is that, of course, we know a lot about the effects of these. We know that Rosier and Eluta together are kind of co-dominant, and Sulfuria is dominant in a certain direction. That makes predictions about which ways we would see deviations from our simulations. But there's a danger of falling into storytelling. It's not clear to me how to sort of rigorously use the information we have about the actual phenotypic effect. And of course, it shouldn't be us doing the judgment. It should be the bees doing the judgment. But that's yet another enormous life's work, I think, doing selection experiments with bees. Another issue that I've kind of skirted around is the SNP markers are most likely not causal. They're our best estimate. They're the markers that are kind of best correlated with flower color. But they won't be actually at the causal locus. So there will be, some of this step pattern will be due to introgression, in the sense that if the underlying Klein is like that, then a tightly linked marker will kind of hitchhike in the middle. It's kind of spatial hitchhiking. So we'll see this step pattern. And some of that is going on, and that's the sort of next stage, but we're hoping we can write up this stage of the project without getting into that. We have another sort of project on Kleins along the genome, which we have data on. And it makes sense, you know, if you take the marker that's closest, it looks kind of sigmoid-ish, or with a mild step. As you go further away, the step gets smaller and smaller. So we actually have markers going away from Roselle, which are sort of doing something like that, roughly. So we need to analyze that properly. Finally, as I've sort of hinted several times, you know, we go in saying, oh, it's all about the bees. We don't have direct evidence for that. We've done quite a lot of work trying to get to it, which is always on the margins of significance. If you sit looking for the bee to pollinate, it's very boring. Every half an hour a bee comes and maybe it goes in, maybe it doesn't, and then you film it. And then we've avoided this. We realized it was not, it was going to drive us mad. But there's a group from Toulouse, Monique Bruce and Christophe Andelo have spent a lot of time on this, filming flowers. And then you've got to watch the videos. It turns out AI just doesn't work with, flowers waving around in the wind. And their solution was undergraduates in practical classes who had to watch the videos. But, it's really hard to get the sample size. So then Rico had this idea, which he called the cigarettes, the Humphrey Bogart type thing, where you put little discs into the flower. When a bee comes in, it falls out. So you can see if a flower's been pollinated, and that gives hints of selection, but it's all very difficult. What we have most hopes for is something we're in the progress of doing. We've taken fruit capsules and grown up seedlings from those. We know the mother, we can infer the father from the genotypes, and therefore we get the father's fitness. And that has to be something to do with pollination. So at least we'll be able to say, is it about paternity? Is it about pollination success? Finally, the most disturbing thing, and maybe the most relevant to us here, is, well, how much variation is actually captured by all this population genetic stuff? There's classical genetics of major loci affecting flower colour. Of course, one can pin this down, but if you look at the flowers, they're very, very variable. No two flowers are the same. And it's hard to even know how to measure, how to define this as a kind of high-dimensional trait space. We've done it with various sort of schemes of looking at the distribution of pixels of different colors in different landmark regions. So you can choose a number of homologous regions and then do the distribution of pixels and do principal components and all that sort of thing. It's a lot of dimensions. I mean, how do we actually ask or even phrase the question, how much variation in some sense is captured by these 7 loci as opposed to other stuff? And is there other things going on? No, it's not just about flower color. I don't know how much these antirrhinum care about flower color. It's a big part of their fitness, but not everything. And I would be amazed if there wasn't adaptation to drought, to altitude, to people coming along mowing you down with a strimmer, all kinds of challenges they're facing. But would we see that? If it's truly polygenic, how much of that kind of adaptation is going to be reflected in the kind of genome scans we're doing? That's it. Plenty more work to do. Yes.
So if you look at your selection patterns.
So if you look at the selection patterns, from, if I understood correctly, where you compare the ones that. So if you look at the selection patterns, from, if I understood correctly, where you compare the ones that give raised to offspring, those ones that didn't, is there seasonal variation that you find, or yearly variation that you see? It varies between the years, that you find in some years this is higher, the other not?
Haven't looked yet. Yeah, there are an alarmingly long list of things like that to look for. I mean, local density is one thing, size of plant, all these things. Yeah. And of course, you sort of run out of power fairly quickly. So, frequency dependence, all I'm doing is saying, well, are the magentas doing better on this side and worse on this side? One can do that. Breaking it down into little patches and seasons and years, probably you lose power. So. And this is, even though we're sampling a big fraction of the population, maybe we can do better with the new data set where we've got more years, more individuals. We can fill in the pedigree, we can go beyond trios. That will help, but it'll always be incomplete, I think. I have to stop.
Can I ask one last question? Can you, so at some point you said that you could estimate, with the correlation between loci, you could infer the dispersal. I don't quite understand how that would be independent of the selective coefficient. It's because you use the same loci that are in the selection to infer
That's
it.
true.
So can you help me sort of understand that?
So it relies on some theoretical work, really. So if you have a cline maintained by selection, some kinds of selection will not have much effect on LD. So if it's selection against heterozygotes of two loci or selection, frequency-dependent selection acting independently. If it's epistasis, then depending on the direction, it will increase or decrease the LD. So it will have an effect. However, regardless of selection, there's always this mixing. And it turns out that mixing just numerically has a larger effect than selection, typically. You have to, I mean, the point is, if you put in really, really strong epistasis, which generates strong LD, then you get a really sharp Klein, which generates even more LD due to mixing. So it's really a sort of a rough approximation that says, let's ignore the selection and just look at the contribution from mixing, which in the simplest models, of independent selection with no epistasis would produce a simple relationship, and it's not far off if you have different kinds of selection.
So Nick, I think your study is basically unprecedented in scale for a natural population. So what, if I would like to study a hybrid zone now, what would be your recommendation for me to do, how I should design my experiment that I get most out of the money that I have? Probably will be much smaller financial scale, but what can I learn from this in designing an experiment that would be suitable for another hybrid zone?
Yeah. So I'm obviously not saying everyone should do 30,000 individuals. It's kind of mad. That's because we wanted a pedigree, that's why. You do need quite a lot, though. I mean, there's a lot of Klein studies which do, 50 individuals, and it's kind of, a lot of it depends whether you're in one dimension. If you're in a really linear transect, maybe you get away with it. And of course, in the beginning, I mentioned this genomic Klein thing, the whole genome data, we had 8 samples, so it was kind of really extreme, but that really wasn't enough. If you wanted to, I guess, I would say, if it's a decent one-dimensional transect, a few hundred, you're doing something decent. The Bombina was actually, for those types, quite a really big data set. That's why we got really good estimates. It was a few thousand, so that was on the big side. You don't necessarily need many markers because, you can get a lot out of just a few markers, and, if you get more markers, they're all linked with each other. We are now looking with the haplotype data, of course, at the genealogical structure in small regions, but it's a whole different enterprise. It's about the deeper history of the thing. The short-term stuff, all the Kleins are really equilibrating in the short term, which is a strength of the system because it means they're quite robust to all kinds of demographics, stuff you don't know about. But because it's short term, it means you don't really gain anything from having lots and lots of linked markers. That's all telling you about deeper history and longer time spans.
So if I summarize it, you would say I should basically do what you did in the beginning, do a rough estimate of decline, do pulse-seq or whatever, look at allele frequency differences, then I pick my markers, and
then you're fine. And the main thing is not to measure dispersal, because the reason most Klein analyses come out with convincing kinds of, plausible sounding answers, and they get estimates and they all sound good, is that they just guess what dispersal is. I mean, if you look at most things, they just say dispersal in this organism is 50 meters. And you look back and it's someone saw a butterfly go 50 meters. It's something completely, because it's really, really hard. There are hardly any situations where you can really measure dispersal. I mean, I have to say I was lucky in my first PhD project because I did wingless grasshoppers, brightly colored, alpine meadows, short grass. You can just see them. You write numbers on them and you follow them, and they follow a normal distribution. They're really stupid. They do a random walk. And so you can measure dispersal, but, birds and so on, you can get something if you have enormous ringing efforts and so on, but it's super hard. And what we were surprised by actually in this whole antirrhinum is that the leptokurtic dispersal is so extreme, it really dominates the shape of the Klein, which I'd always thought, well, diffusion is a pretty good model, but diffusion at some point breaks down, and it breaks down here.
So you're saying basically you needed a large data set to estimate dispersal reliably if you cannot observe it like...
In most cases, you just can't do that. You can only, I think, estimate dispersal either by direct observation, which means you have to do birds and have lots of ornithologists, out there, or you do a pedigree of some sort.
Do you have variation in seed size that could sort of create variation in dispersal?
Actually, we're looking at that. I don't know. I mean, there is variation in seed size, although they're tiny seeds, so it's a bit hard to measure. But
But if
I
they're
don't know how you
like
correlate it.
relatively heavier, they would fall more
Yes, but
locally.
the trouble is you don't know the size of the seed that led to the plant that you see. So I don't see any way of, unless we made radioactive seeds, but we're not going to get permission for that. I mean, I don't know. Yeah.
So have you looked for, or do you have evidence for the evolution of the cline itself, either in the steepness or in the position?
Poverty, actually, you did look at this. It didn't seem to have moved. No, I mean, but if you only have 10, 12 years, it's not, you don't expect to see much in that time. The only two lines of evidence. One is that there is a vague report of some hybrids from somewhere in that region from 1930s, right? So, yeah, I mean, so, okay, similar. It can't have been there 10,000 years ago because it was under ice. A thing that's a little bit better is that you can look at recombinants between Rosea and diluta, and there's half a centimorgan. So every generation, a certain number of recombinants is generated. So there have to have been, if they came together with no recombinants and they started recombining, it needs at least 100 years to produce the recombinant level we see. So it's probably older than 100 generations, actually. Now we know 100 generations is 300 years. So, but we don't have any very strong evidence. I mean, I'm kind of hoping that it's been, it didn't just arrive a year before we started studying it. That would be it.
Yeah. Nick, you may have mentioned it. How stable is that inflection point of the decline over time? Does it stay there? Is it kind of wander?
It's stable. Yeah, it seems.
That's kind of odd.
Is it?
Well, I mean, just drift alone would push it because your sample size is
Okay,
pretty
so
small.
first of all, if you do the calculation for drift, declines have a lot of inertia. So given the population size we have, actually NE is 15,000 in this region. That's because the seed bank is inflating NE. So there's a lot of inertia. Yeah. And the other thing is, what was I going to say? Can't remember. Yeah. Okay.